to research rounds. So I'm honored to present and welcome Dr. Shuhendler to our hybrid event. Just to remind the audience members here, for those in the room, if they have any questions, to use the microphone over on your left. And for those joining virtually, submit your questions using the Q&A, not the chat feature, please and thank you. And we'll also be accepting live questions at the end, so just use the hand up feature and we'll unmute your mic so that you can ask your question directly. If you're experiencing any difficulties, you can use the chat feature for that. So Dr. Shuhendler is going to be presenting today on towards imaging-based patient stratification for interventional cardiology and nephrology. He's associate professor at the University of Ottawa and holds a Canada Research Chair in chemical biology. He holds a primary appointment in the Faculty of Science and Department of Chemistry and Biomolecular Sciences, and is cross-appointed in the Department of Biology and holds a scientist position at the Heart Institute and also affiliated with the Brain and Mind Research Institute, where he's a member of a concussion injury group. His research focuses developing new imaging agents across a range of clinical modalities, including MR, PET, and OCT, to improve our ability to obtain patient-specific information about disease or injury, as well as a patient's response to therapy. He is a recipient of the Killam Fellowship and a co-founder of two companies, including the Echidna Sensing, advancing rapid tests for cannabis quality, and Yellowbird Diagnostics, very recent spin-off looking to commercialize imaging probes developed by his group. I think his work is very relevant to the cardiovascular field. He's been doing a lot of work, as I mentioned, with OCT and with imaging that we're excited to be able to investigate and potentially apply in patients, some of his new discoveries, and really look forward to his multi-specialty presentation today. Adam.
Thanks so much for the introduction. Thanks for being here today. So I'm going to talk about two stories from the lab. We've been working on them for about 4 years now. And one works on IVOCT for cardiology evaluation. The other one is MRI for evaluating kidney function. So I just want to... disclosures, so co-founder of Echidna, which is less related to the talk, but two of the technologies that I'll be talking about are being licensed to Yellowbird, which I'm fortunate enough to have some senior members of the lab are going to take over and try to spin this out to commercialization. So just an overview of the lab. We kind of operate under sensing for good. So we try to think of ways to apply technologies for sensing, mostly imaging, but maybe some point of care thought too, where we start with innovating new sensors, so chemical sensors, nanoparticle-based sensors, for biomarkers that we're told are important or that might be important by the clinicians we work with. We apply these in vitro and in vivo preclinically to try to learn more about some of the diseases and tissues that we're evaluating. But ultimately, our goal is to make things that can get to patients if they show value along their road of development. So this is the most recent picture of our group. And so the work today has been mostly done by Nick and Alexia, shown here. But everybody in the group has contributing to a whole number of projects. I'm losing count, which is probably a good thing. So the way we work in the lab is we find a need, and we don't necessarily know the need, but we ask a lot of people about it. So some of the clinicians that are in the Ottawa area get some random weird emails from me every once in a while about what can they use to make their lives easier and make their patients healthier. So we identify the need, find a molecular target to fill that need, and we kind of have this multi-tool of modalities that we're interested in. My parents used to say as I was a kid, I never played with one toy for more than a few minutes, and it's probably why I can't stick on one modality for too long either. But the solutions are interesting that way, and it keeps us on our toes as a lab. So just before we go into the data, I want to just define the scales of imaging. So we're all familiar with anatomical imaging, like an X-ray, where we can see structures of tissues, in this case, hard tissues like the bone. We have functional imaging that can be used to extract how a specific organ or cells within that organ are working on physiological processes. And then we have molecular imaging, which kind of maybe looks a little bit like the blobbiest and less exciting of all of them. But I think it's the most information rich, because now this tells you about where and how much a specific biomolecule is expressed and functioning in the body. So we can get really down to those single proteins, single metabolites, map them, and see what's happening during disease. So today I'm going to talk about two stories, one in cardiovascular disease and one in kidney dysfunction. These were both needs that were given to us, one by Ben Hibbert, who challenged us with molecular imaging for IV OCT, and the other one by Dylan Berger over at the Kidney Center, saying that we don't do a very good job of stratifying patients for kidney functional risk. And so we looked at these two challenges, and we came up with markers and how to evaluate them. So the first one was we're going to look at P-selectin, a protein marker for inflammation, using IV OCT with a nanoparticle-based solution. So I guarantee that everybody listening knows more about these numbers than I do, but cardiovascular disease is a big problem in Canada. Lots of people suffer from it. We can even talk about how many people per hour will get diagnosed. So this is a massive issue. And the problem we're looking at is atherosclerosis or plaque buildup in coronary arteries. And so currently medicine kind of waits till this point here where we get a large plaque, which might already be ruptured, before we really can do a whole lot of intervention. But this is very late. And in this really nice article in JACC, they're saying that we need to start looking at these subclinical stages of atherosclerosis, start stratifying patients here and treating people at this point so they never progress to plaques and rupture. So this is wonderful, but how do we see plaques forming before plaques form? And this is where molecular imaging comes to play. What I wanted to also do, because I didn't know anything about IV OCT before Ben challenged me to this, basically they feed catheters up through your leg into your heart, and the catheter has a whole bunch of ranges of balloons and tools on it, but also an imaging component. And so optical coherence tomography, or OCT, it's like ultrasound imaging, right? So instead of sound, it uses light. So it bounces light off of the tissues, and the light reflects back, and whatever that detects, it can reconstruct an image. And so this is really done with this tiny catheter that has a 1350 nanometer laser that rotates within your vessel and creates an image of the wall. And so this is done every 3 minutes or so in North America. It's very common to do it while placing a stent, right? So a stent is where a mesh is placed into your artery where there's a plaque. A balloon is inflated to open the artery, and the mesh keeps it open so that you get more flow. And so this, again, is a very late stage disease. We can see the plaque. We can see the anatomical signs of plaque, but where do we stop treating? Right? How far does that plaque go? How far does the inflammation stretch around that disease? So this is what an OCT looks like in the clinic. So this is your vessel wall, and this is the lumen, the big black area. Usually in the clinic, they flush it with iodinated contrast agent, not to produce signal, but to clear the blood cells. So that's why there's nothing in between. And then this circle on the inside, this is the imaging catheter. And then sometimes you'll see this kind of smiley face with a big shadow. This is a metal guide wire, right? So this causes regular artifacts. So what can we see already with IV OCT? Okay. Oh, I forgot about this. Sorry. So again, uses a laser. So it's a laser light that scans from the catheter out to the wall, and it rotates quickly, and that rotation causes the image based on the laser light that's reflected back to the imaging head. So we can see thrombi, right? So anything anatomical and morphological is really easy to detect with IV OCT. So red thrombus versus white thrombus. So here you can see with the red thrombus, you get this big shadow, probably because of the metal content within the cells, whereas the white thrombus has less of a shadow behind it. These little spokes here are from a stent, right? So the individual pieces of metal, they create this reflection on top and the shadow on the bottom, right? We can also see plaque. So here's an IV OCT image. Again, this smiley face is your guide wire. And then this is a cartoon showing the anatomy of the plaque. So you can actually see a lot of the information, but this is very late stage disease, right? So how do we do this earlier? How do we get earlier information about inflammation? So we're aiming for this no atherosclerosis phase, right? And so we want to see two things. So one is we want to identify patients that have vulnerable plaques, because those are the ones that need to be treated rather than the ones with stable plaques. And this can be done decently with IV OCT. We can see thin cap fibrous atheroma. We can see invaded macrophages. We can see thrombi. We can see neovasculature forming on the plaques that show it's becoming more mature. So again, lots of anatomical changes can be seen, but this is all in this late stage disease. So there's currently no way of using IV OCT to see these pre-anatomical changes, right? So molecular changes are still invisible to this imaging method. So this is where we feel we have a place. So we thought about what do we need in a contrast agent to be able to be effective and to be used clinically, right? This is one of our goals that I started with, is we want to make things that have translational potential. So they obviously need to be biocompatible. They need to fit into the clinical workflow. And again, this is the clinical imaging catheter. So this winds through the femoral artery up into the heart, and your imaging head is at the end here, and there's a flush port. And this is how I was talking about iodinated contrast agent was flushed in to clear the blood cells. So we want our contrast agent to be flushed in through the same port. And this allows local delivery of contrast, but also no change to clinical protocol. We just swap a syringe instead of having something else that has to be incorporated. Obviously, we have to interact with the OCT light, which is 1350 nanometers, which is in the near infrared 2 window. High signal to noise, because we probably have very rare binding events, right, relative to the size of the artery. And we want scalable synthesis, okay? So we thought about this stent signal, how you have this flashy line and then this shadow, and it's kind of like a 2-for-1. So we thought if we take the 2-for-1 of a metal, how it interacts with the IV OCT laser, and turn that into a contrast agent, we might be able to really see very rare binding events. And so we needed a metal that was biocompatible and tunable photophysically, and the first thing we thought about was gold. So there was a paper published in 2015, I believe, this one here, where they took gold salts with a reducing agent and a capping agent, and they were able to heat it in an oil bath, form particles that then clustered together. So these are clusters of nanoparticles. And what you can see over time is the solution goes from an orange color of the salt through the assembly of the gold particles, forming a black solution of the clusters. And so black means it's absorbing all the light, right? And we thought, well, okay, they didn't go all the way out to 1350 on their scans, but they went to 1000 and they had a really high signal. So let's see what happens. The other issue, though, with this method is they couldn't stop at spheres. They had to just keep heating, and you ended up getting aggregates of the clusters, right? And so when they actually model their spectra, they have all different shapes of clusters present. That's not great for predictability and reproducibility of synthesis. So we kind of took a part of their method and optimized it. and we used a microwave. So we still took gold salt with their capping agent and reducing agent, and we microwaved it. And our first microwave solution turned black in about 5 minutes. And under TEM, you can see we get these clusters, and this is a zoom in of the side, and you can see all these individual nanoparticles making up that cluster. The problem is we see lots of different shapes. So we thought, well, what if we clean them up and hold them together with some polymer? So we wrap them in polymer once, and you can see already we're getting more spherical clusters, still with those individual particles making them up. And when we did two wrappings, you can see we basically ended up with these spherical beads. So now we've overcome that spheres of sphere problem they had, and we can have spherical beads based on gold nanoparticle clusters. So this is a really high resolution SEM of our clusters. And we have this cool facility on main campus that has a helium ion beam that can basically cut what's in your SEM. And so they were able to basically core our particles like you would kind of slice into an apple. And we can see that they're actually solid, right? So they're actually all contained of or comprising gold nanoparticles. We sonicated these to break them apart, and these are the individual gold nanoparticles that make up our cluster, and you can see the distribution is actually quite tight. And again, this is all done in a microwave. One pot takes about 5 minutes, so scalable microwave synthesis, you can scale up to 100 milliliter or higher batch sizes. So what about the photophysical properties? So here, just on the top, this is size measurements in solution. And you can see as we wrap our particle in polymer, we go from the light blue, which is unwrapped, to the dark blue, which is double wrapped. And we get a really nice, relatively tight distribution, considering we're making these superclusters. And the best part was the dark blue line, after we wrap it twice in polymer, our peak of interaction with light is almost exactly at the OCT laser line. So now we have material made of gold, which we know is biocompatible, that can interact with OCT light. And when we separate the absorption and scattering, we can see that at 1300 nanometers, it's mostly scattering. And this is great because that's how you maximize your response in OCT, because you basically prevent that light from hitting back to the receptor, the detector, sorry. So here we have scalable materials that can interact with OCT laser light, and we have scalable synthesis. So what's next? We want to see the contrast properties. So all of our evaluations that you're going to see are done on a clinical OCT instrument from Abbott, the Illumian, using a Dragonfly catheter, which is also the clinical standard IV OCT catheter, at least in use at the Heart Institute. And what you can see, so we filled a glass tube with our particle suspension, and this is the pullback, an image of the tube. And so we start at 730 microgram per mil, and even at 46 microgram per mil, we still have good signal over saline, which is shown here. But what's really important is we compared this to individual small nanoparticles, as well as large gold nanoparticles the same size as our cluster. But this is just a single monolithic particle. And so the small nanoparticles at the same gold concentration as this phantom here, you can see almost no signal production. And then this tube here is the signal production from nanoparticles that are 500 nanometers in diameter. But these are monolithic, right? So same size as our cluster, but not the same photophysical interactions. And actually, we end up getting almost threefold more signal from our clusters than we do of the equally sized gold nanoparticles. So this is because when you put gold nanoparticles together, you get something called plasmon hybridization. And this is how the electrons on the surface of the gold particle interact with light. And when you bring all these particles together, they kind of fuse their plasmons. So by bringing all these gold particles into a cluster, we fuse these plasmons, we've extended our pi conjugation basically, and now we can interact with near infrared 2 light. So I challenged my student before we entered animals to do a pullback image. So the way OCT imaging works is you put your catheter in, there's an automated pullback system that pulls the catheter at a given speed back through the vessel so you can generate your image. So he made this tube phantom. This is just a polypropylene tube filled with agarose gel. And in the agarose gel, he suspended our clusters or individual gold nanoparticles and did a single pullback. And you can see the cross section of that is the only time we had a real signal increase was when we pulled back through the gel containing our superclusters. So here we have high signal to noise contrast, which is obviously very important to advance to IVOCT. So what's next? So I started the talk with a lot of looking at the invisible, right? So this is now we have a platform based on a high contrast cluster. How do we target that to a molecular biomarker of interest? So we chose P-selectin, maybe not because of its clinical utility, but it is certainly easy to access, and it's a well-known early marker of inflammation that's expressed on the vascular lumen. And so P-selectin is basically the receptor to prevent leukocyte rolling past the inflammation. It actually hooks on to the leukocyte and kind of keeps it in the vicinity of the inflammatory signal. And the ligand for P-selectin is sialyl X. We're not making this. We will never make this. This is not worth making because it's not scalable. But one of the key sugars is this one here, is fucose, has this methyl group. And there's papers showing that if you bind fucose and sulfates to the surface of particles, they will mimic sialyl X and bind to P-selectin. So we took a shot because that was a little simpler. So this is the polymer we use to wrap our clusters with anyway. So it's a copolymer that we actually eat a ton of every time you wash your hair. You know, a little bit of shampoo goes in your mouth. This is what it is. This part sticks to the particle, and this hydroxy group gets presented to the outside. So we thought, well, we can activate that hydroxy group and then just synthesize these ligands with a terminal amine that will react quite easily. This is not difficult chemistry or new chemistry at all, but again, scalable. And so we made fucose, galactose, and sulfate forms of our polymer. And we just mixed them together in ratios as we did the coating. So we end up with our cluster presenting different ratios of sugar and sulfate on the surface. Assay in vitro, we coated a high binding plate with P-selectin or albumin, and we incubated our clusters, and we shook it because if you have to keep the particles under motion, otherwise they won't properly attach to P-selectin. And what we found was that relative to bare particles, we had a significant increase in gold retention in the wells when there was P-selectin present. And it didn't really matter if we used fucose and sulfate or galactose and sulfate, and the ratios, we got really good binding. So the nice thing, though, is that the change in the surface doesn't really impact the photophysical properties. We still get that peak at 1300, and they still look like nice round beads on microscopy. And so now we showed that through this functionalization of the polymer that we use anyway, we can bind disease biomarkers. So this was all in vitro up till now. And up till now, I talked about the synthesis like we just put it in a microwave and 5 minutes later it was done. The students spent about 6 to 8 months optimizing that. It's a table with I don't know how many entries of different mixtures. And so the fact that we got to this point is pretty, I was really happy with. So now we wanted to do something in vivo. So in healthy vasculature, this is our imaging probe, we'd have no engagement of the target because there's no inflammation. And so what we'd end up hope to see is just the wall and then our catheter and nothing else. What we anticipate is that when we have inflammation, we'll get P-selectin expression, we'll get engagement of the P-selectin by the cluster, and where these clusters are sticking, we'll get the similar imaging outcome as we would with a stent strut, right? So we'd have the buildup of metal in that area where we'd get both this bloom, this line, and this merry-go-round, which is the shadow that forms behind it. And together we call these roses. So when we see these in the context of molecular imaging, we call these reflection or scattering events because they're not artifacts anymore. These are intentional signaling production, and we can actually count the number of roses per section and correlate that to disease severity, which I'll show you. So we wanted to stay on using the clinical infrastructure to do this evaluation. And the catheter is much too large for any vessel in the mouse, and it just fits into the abdominal aorta of a rat. So we were able to generate an intravascular inflammation model in rats. So we gave some LPS, so this is bacterial cell wall lipopolysaccharide IV, and within 4 hours, we get luminal expression of P-selectin. So we were able to expose the abdominal aorta in the rat and then insert the Dragonfly catheter and go through a clinical workflow of IV OCT. So these are the results. So we have before we injected contrast image on the top row, and then after we did the injection of contrast and saline flush to clear contrast on the bottom row. The first column is LPS treated animal, so an inflamed animal with our non-targeted cluster, right? The middle one is our diseased animal with our targeted cluster. And the last on the right is our an untreated healthy animal with our targeted cluster, okay? And so what we can see is that we have very little targeting signal rose production in the untargeted clusters. Here you can see the roses in the sick animal with our targeted clusters. And then we have no rose formation in our healthy animal. And so we quantified this roses per centimeter, and you can see a significant increase when we have targeting in a diseased animal. And we scored the disease severity of the abdominal arteries, and we can see a decent correlation between the number of roses per centimeter and the severity. And just so you can see, so IV OCT is actually a 3D method. So these are the real-time pullbacks that you see. And so you can see there are some roses that, that do form. This is again the healthy, the diseased animal with the untargeted particles. But you can see by far the most roses are forming in the middle animal, which is diseased with targeting, and nothing at all in a healthy animal with targeting. And so these were quantified as they would be evaluated in the clinic, right, through axial cross sections like this. So this was all really exciting. Again, this was the sum of 4 years or more of work. But basically, I think we've shown, at least preclinically, we have a biocompatible contrast agent platform now for IV OCT molecular imaging that fits into the clinical workflow. So again, we just injected these into the imaging catheter, let them sit for about 30 seconds, flushed, and then imaged. So we didn't have to do a 24-hour incubation or anything. It really fits into what's done. They interact with the laser light, give high signal-to-noise contrast. They bind at least P-selectin, but I'm sure we can target these to other molecules as well. And again, scalable synthesis. And what I didn't show was we actually did a pilot project funded by the Oracle funding, the precision medicine funding, in 2 pigs. So these were the big full-size pigs. And they each got 3 imaging sessions over 24 hours. There was no issues at all with the pigs. But prior to that imaging, we had to sterilize, and we did that by gamma radiation and didn't affect the particle at all. So towards clinical use, we were starting to check some of the boxes. So I want to switch gears now a little bit and move from heart to kidney. And we're going to move from IV OCT to MRI and find a way to map glomerular filtration rate on the kidney itself to try to really pinpoint what's happening in patients with kidney dysfunction. And so kidney disease is a global health problem with more than 13% mean global prevalence. And GFR is the gold standard for kidney functional measurements, right? So just in case people forgot, because I certainly did before starting the project, you exchange from your blood to your nephron through the glomerulus, before it enters a loop of Henle, where it just determines if it's going to be reabsorbed or excreted as urine. So this is sort of the first point of filtration that's very important for maintaining blood toxin levels. So currently, creatinine is used as a blood test to measure GFR. So you measure an individual patient's creatinine level, you put it through a population-derived equation, and you get something called estimated GFR, eGFR. 20% error rate is acceptable based on these measurements, which is quite high. And there's also different corrections that are required to be applied based on patient ethnicity. And that adds more error now on top of it. But even with these measurements, you get a lot of unaccounted for characteristics, like what's the cause of chronic kidney disease, which can differentially alter creatinine levels. And sometimes there's just steady state changes in creatinine levels from patient to patient that doesn't get accounted for. So there's a need to properly evaluate kidney function. So there are ways. Some are anatomical with CT or ultrasound. There is a SPECT method to evaluate kidney function, but the resolution of SPECT is quite poor and semi-quantitative. And so the actual, it's actually more error prone than creatinine-based EGFR evaluations. We talked to Dr. Nick Sciera, who's a body MRI specialist here at TOH. And he said, just do it under MRI. MRI is a one-stop shop for the kidney because you get anatomical, and if you have a contrast agent that can map function, you can get both in one session. So the idea was that we would give a contrast agent and take images over time and see how fast the kidneys clear that contrast agent and try to relate that back to glomerular filtration. So this would give us multi-parametric evaluation with high temporal and spatial resolution and non-ionizing radiation as well. The problem is current MRI contrast agents are contraindicated in patients with actual or suspected kidney problems. And the issue is that they're gadolinium based. And when gadolinium accumulates in tissues, you can get something called nephrogenic systemic fibrosis, which is a systemic fibrosis formation that's quite painful. And this, again, can happen more likely in patients that can't properly clear the contrast. So we had to find something else. We couldn't use gadolinium. So the solution was Free Willy here, basically. And organic radical contrast agents, or ORCA, were previously reported. They were focused on this molecule called TEMPO, and this is a nitroxy radical. But when we applied basically a very mild reducing condition that's present in the body to TEMPO, you can see we lose the signal very, very quickly. So we looked through the literature some more, and we came across this verdazyl radical. So you can see this is a nitrogen-centered radical, but because there's lots of electrons around that ring, it gets shared amongst all those nitrogens. And because it's shared, it's more stable. And so by forming this verdazyl radical, you can see under the same conditions we incubated TEMPO, we have no loss of signal. So it's a very stable radical. So we gave this to mice. And what we noted very quickly was that this is your pre-contrast injection. So here are your kidneys, and if you look at muscle as a comparator, you can see we get an enhancement in the kidney within 2 minutes of injection and complete clearance by 40 minutes. And there's no uptake in the liver or the muscle or the lungs. Really, the only thing we see uptake is the kidney, which clears down to the bladder. And so we can track this over time. And what we get is if we take we have a decay constant that we can take from the semi-long plot of the signal over time. And you can see we get this really nice straight line of decay. And this is actually over 9 mice, which is incredible to me. I mean, in the error bars I've never seen like this with an imaging agent. And so here we have a non-metal contrast agent that goes into the kidneys, clears into the bladder. We get no uptake in any other tissues, and we get a very, very strong linear regression of decay in animals. So we thought, okay, this is great. We're going to do slice-wise imaging of the kidney and do something called dynamic contrast enhancement, which is done in the clinic regularly. We can look at different regions of the kidney, so the medulla, renal pelvis in pink, and then the cortex in yellow. And then we can look at the change in the overall kidney intensity over time, or we can do pixel-wise maps. So each of these pixels or voxels in this image, we can actually map the clearance rates in that voxel and see what's happening in the kidney voxel by voxel. And so we did this in 2 mouse models. One is called the unilateral ureteral obstruction, or UO. And this is where we go in, tie off one of the ureters, and this causes hydronephrosis. Basically, the kidney can't clear what's in it and kind of explodes. And then we had our control animal where we did the surgery, but just touched the ureter instead of tying it off. So when we measure the mouse blood creatinine levels, we can see, as we expected, in the ligated ureter, we get an increase in creatinine in those animals. So we certainly have a nephrogenic response. And this is the imaging. So the top row, these are the T1 weighted contrast images. So this is the ligated kidney. Obviously, there's something wrong. So we don't need the beyond anatomical to say that kidney is not working. But the bottom is color-coded for clearance. So the darker the color, the higher the clearance value, and the more red the color, the poorer that kidney is functioning. And so in the sham animals, what you can see is fairly consistent functioning over time. But when we do the obstruction, you can see right away the obstructed kidney stops to work, right? We get accumulation and no clearance in that tissue. This is verified histologically. Again, you just get buildup of fluid in that kidney. But what was really interesting is the ability to map the clearance rates across the kidneys. We were able to see decompensation in the contralateral kidney. So the one that wasn't ligated still obviously has some effect. It has to do all the work now for 2 kidneys, and it started to fail as well. So even when there's an obvious, one of the kidneys is obviously damaged, we can use this agent to evaluate how well the other kidney is still working. But this was a bit extreme. So we now move to a model called folic acid-induced nephropathy. And this is where you give the mice a lot of folate. It accumulates in the kidney and crystallizes. And that crystallized form of folate starts to really cause fibrosis. And so what we can see again is as we induce fibrosis, we have an acute phase at 15 days and a progression to a chronic phase at 30 days, and we have an increase in serum creatinine levels. So when we did imaging, again, showing the maps, so this is the healthy kidneys. You can see here in the acute phase, we get significant changes to the clearance rates in the medulla. But even in the cortex, you can see we're getting striations of almost no clearance function at all. And then at the chronic phase, it seems to get better, right? And this is actually well known clinically, is you have this progression from acute to chronic. And so our renal decay time constants, right, the slope of that curve gets really bad at 15 days and shows some improvement at 30. But this is counter to what we see with creatinine, right? So creatinine just keeps increasing. So how do we reconcile this? Well, if we look at the histology, this is trichrome staining. So the blue is fibrosis. And so obviously at the 15-day acute phase, there's fibrosis in both the cortex and medulla. And that's what we see on the imaging. We see loss of function. But at day 30, we do get some regain in function, and you can actually see we get a recovery phase from fibrosis in the cortex and a redistribution of fibrosis in the medulla. And it's that redistribution and recovery that we're actually seeing on the scan that's not taken into account when you're just taking blood creatinine levels. Is GFR yet, because we don't know if it's GFR. We know that there's a linear decrease, but is it actually GFR? So there's this company called MetaBeacon, and they sell these tiny spectrophotometers. They're actually a spectrofluorimeter and a battery pack. And these get taped onto the side of the mouse, and you get constant readings of subcutaneous fluorescence while the mouse kind of runs around in its cage. And they've done this and shown that if you administer this sinisterine, it's a fluorescent peptide that they've shown is 100% GFR cleared, you get these curves that form. So every dot is a reading that spectrofluorimeter takes as the mouse is running around, and you can see its clearance over time, and we can extract the GFR rate. And what we see is that on day 0 is the black line. Day 15, the acute phase, we can see a loss in GFR, and at day 30, even though the curves overlap, the slope is a little bit better. And this is our MRI data. And actually, when we plot the GFR derived from the transdermal kind of gold standard against our MR decay, you can see there's almost no difference. So we think that our actual decay constant that we're measuring in the kidney is actually GFR. And we can actually map GFR now by MRI. And again, this is a metal-free contrast agent, which overcomes that contraindication to the populations that would need this evaluation the most. Basal, we've now kind of achieved this precision renal medicine or safe MR urography, where we can look at not just individual kidneys, but individual voxels in a kidney and see what's happening over time. And it seems to correlate quite well to diseases, at least in mice. And we're hoping to move this forward. So I was asked in the last couple of minutes to talk about something to do with EDI. So I'll admit that we don't do a whole lot of big animal studies, so we don't usually compare males and females yet. But I guess my dedication to EDI comes more in outreach. And so in the Faculty of Science, we have a couple of programs we've been working on for a couple of years now in terms of Indigenous outreach. One is in concert with Let's Talk Science, and they have a new Indigenous mentorship program where they pair a professor to a community and hope to build a relationship of interaction between that professor and high school students in that community. I've been fortunate enough to be assigned to the Moose Creek community, Moosonee and Moose Factory, and we designed different programs for them. So here, what you can see is this is a traditional snare that they use for trapping. And we did a whole series on materials properties that could be used for snares, and talked about malleability, brittleness, different material components that go into snare building, and went up. And unfortunately, I wasn't able to go because of teaching duties, but this is the snare demonstration that we built, and they took it up to Moosonee and were able to do the activity with the students. And most recently, within the last 6 weeks, the same class was able to come down to Ottawa, and we introduced them to gold particles. So we had them make little gold nanoparticles with the Turkovich method, different colors of particles, and then we showed them how to make it into paint, and they were able to paint these kind of window dressings, but we talked to them about nanotechnology, and they had a hands-on experience that way. And then within our faculty, we started something under Jacqueline Brousseau, who's the Vice Dean of EDI and other things. I don't remember her official title. If you're watching, sorry, Jacqueline. But we have a new program called UOASIS. It's Ottawa Advocates Supporting Indigenous Science Students. And so it's a group for all students in the Faculty of Science, from undergrad to grad, to come together and just talk, meet profs, find out about what's next, how to succeed. And through that, I initiated a program called the Pathways Program. I convinced our dean and the OVPRI to put in $3,000 from the faculty and $2,000 from the OVPR, leaving a $3,000 commitment from the professor to have basically the same program as USRA runs, but focus specifically at Indigenous students in science in their third year, to help prep them for their honors project. So we just launched this program. This summer will be the first time it happens, but we're really excited about that initiative. That's all I have. Thank you.
Fantastic work. It's amazing.
Thank you.
Amazing. And congrats on the OASIS program too. That's also exciting. For the gold study, so you said 4 years to get to where you are now. Presumably you want to move this to humans. What are the next steps for you? And then I have some other questions about the gold.
Yeah, next steps. Next steps are tough ones. So this is efficacy in a large animal model, and then safety. So as a nanotechnology, I mean, really have to be careful. Like so far, we've injected, I'd say 20 something rats IV and have not had any, overt death or symptoms or signs. We've not seen any signs of embolism or anything pulmonary, so we hope we're okay. It seems to clear from the spleen, but within a day, it's kind of gone from all the other tissues, which is what we would expect. But yeah, we need to work on probably a pig model, a well-characterized pig model of vascular disease.
Yes.
Choose a biomarker and do that imaging experiment.
Yeah, so that was what I was going to suggest, the pig, but that you've already thought of that.
Yeah.
And that was my other question, is what happens to the gold? Like, where's it go?
Yeah.
The nanoparticle is presumably smaller than the capillary diameter.
Yes.
So it should pass through and therefore may go to the lung, as you pointed out. But it seems that it's being taken up presumably by macrophages or something in the spleen.
I think so, yeah. I mean, I think the different targeting groups will all, we'll have to evaluate toxicity every time we change the surface of the particle. Yes. But we did do it with the p-selectin targeting on it in rats. And really the only significant change from baseline was in the spleen. And it seemed to peak at 24 hours and then peak at 12 hours, and by 24 hours, it was not significantly different from baseline anymore.
So right now you're doing it with OCT, but presumably because it's gold, you could also do MR.
Not MR, but maybe CT.
I mean CT.
Yeah,
yes,
CT.
Yes.
So we're wondering if it can be seen with fluoroscopy, which would be really exciting too, because you usually combine that with IVOCT anyway, right? So my worry is that the sensitivity of OCT is much higher than fluoroscopy. So I don't know that we're actually injecting enough mass to see with fluoroscopy. But I think that's one of the strengths of using the IVOCT and injecting through the catheter is we're really pulsing small amounts locally, and then it's diluting, right?
Yeah. So that was my other question, that you're pulsing it through the catheter. So you're relying on flow to be able to deliver the gold to the surface. Mm-hmm. And so you're relying on that extraction interface, yes, if you will.
Yeah.
So the flow may change, which may change that rate of affinity.
Yeah, we did use blocking. So we did the balloon occlusion, which is also done regularly and in clinical procedures. So we're able to kind of stop flow for I think 10 or 15 seconds, let it sit, and then resume flow and wash, right? So I think as long as we have that ability to give it that short incubation time to latch on, it seems to work okay.
Okay, excellent.
Yeah.
Well, good luck with that. And we'd be excited to help move that forward for awesome. Sure. Yeah.
Great.
And how about the kidney story? What do you see as next steps for that?
So that's the most. So Yellowbird is moving that forward. We're designing the pre-clinical trial application experiments required to fulfill a full CTA application right now. Yeah.
Excellent.
Yeah.
Excellent. Any other questions? Dr. Chong, I knew he would ask something. Can you come up to the microphone, AY?
So we did imaging in a pig model. So we did it, we sent it to Charles River in Montreal. They have the contract research that does the pig IV OCT. So they did, we did three imaging in 2 pigs over 24 hours, and well tolerated in the pig. We could see the particles not sticking, but we saw them in the lumen, and everything so far was okay. But this is why we need to work on a disease model to get the timing of expression, right, to verify that we can do the binding that we can do in the rat, because the flow is going to be higher a little bit in the pig. So we thought that's sort of the next step.
The flow is higher, and also I think, if I am not mistaken, when you did it with a rat,
right?
How did you put the catheter into the aorta?
Very carefully. No, we had to cut the aorta. Yeah, the femoral is too small.
So one of the reasons might be because your injection and image acquisition is along the blood flow of the mouse.
Right.
in pigs is on the opposite flow.
Yeah, exactly. Yeah. No, absolutely. Yeah, it's one of the issues. I mean, I think the rat model was great for us because we could control it as a first step, but definitely highly limited in terms of the, I think we've done what we can in the rat, right, at this point. So
one way for you to prove on a human model, there are centers in the US who are doing OCT on veins when they're being harvested for bypass.
Oh, I see.
To predict whether those grafts would occlude or not.
Okay.
Say it again. So there are centers in the US who are routinely doing, actually one, because they don't want any other center to be involved, imaging veins when they're harvested for bypass.
Interesting.
So you occlude, you can do that as an animal model, as a human model.
That'd be really cool.
So the difficulty with veins is, so we know the arteries for bypass do very well. The internal mammaries. Okay. The vein grafts don't do as well. Okay. And they kind of arterialize and they develop atherosclerosis. Oh, interesting. So if you had a way to actually predict which veins. Oh, I see.
But you don't get proof of concept as well, right? To see that your P-selectin will actually bind to a human model.
Yeah, that's a great idea.
And the other markers I can think of would be, I mean, I have an interest in angiopoietins, so those are endothelial as well.
Yeah, that'd be great.
We need to meet.
Yes.
Dr. Chan is asking if there are any other biomarkers that would attract and bind the gold particles.
Anything. I mean, any vascular luminal expression that has a ligand that we can put on the surface of the particle, absolutely. The one that we used is supposed to be specific for P-selectin. We didn't evaluate the specificity. We kind of are piggybacking on previous literature. But I think the different animal models we used, the only other receptor that I can think of is E-selectin, but it tends to be expressed at a much, it's more delayed than P-selectin expression. So I don't think it would be interfering at that point.
Yeah, so there's a potential to be looking at different stages of disease as well.
Absolutely.
Sounds like.
Yeah.
Any other questions? All right. Well, Adam, really fantastic work. Congratulations. Really, really, really innovative, and taking, as you put it in that early slide, taking a problem, finding a solution, and basically dissecting it right down to each little phase, the chemistry, and the biology, and so on. It's really exciting work. So
thank you.
Good luck, and whatever we can do to help enable these exciting developments. I look forward to working with you in the future.
I appreciate that. Thank you very much. Yeah. Thank you, everybody.
Thanks, everyone. Yeah.